The next thing that I want to talk to you guys about is recombinant DNA and PCR. You guys have already seen the first part of this, at least a bit about restriction enzymes, and that's good. I didn't cover that in any great detail. This is sort of a first overview look of the technology of recombinant DNA, or what's like sometimes known in the media as gene splicing, though that's not really very accurate. When some people say gene splicing, this is really pretty much what they're talking about. And this is a combination of 3 different technologies which you all know all about now, right? Or well, at least which you all know about now. Plasmids, we talked about plasmid. What's a plasmid? Yeah, how about you? Oh.
It's a small string of DNA, not in the genome, not entirely extremely important to the survival of the bacteria,
but it's separate.
It's separate. Yeah.
It's separate. And transformation. Someone from over here, tell me what transformation is.
It's when the DNA or RNA from the circular one gets split up and then I think it's like the bacteria get infected. I don't know if in that picture.
Sort of. I think you're thinking transduction. That's the one that involves viruses.
So is
you
it
remember?
the one that transfers through?
That's conjugation. It's the one that's not those two. But that's good. Those are both things that exist. What do you tell me? Do you know?
It's when a bacteria picks up a plasmid.
It could be a plasmid or it could be a piece of other DNA. But it's when a bacteria is picking up a naked piece of DNA that's floating around in its environment. And some bacteria naturally do this. Almost all bacteria can be forced to do this. They can be made competent. And the third piece of this puzzle is restriction enzymes, which is what we're going to use to do the actual cutting and pasting. So vectors. You don't just use the restriction enzymes usually to put a piece of DNA into a bacteria's genome. That is possible. But usually what you're going to use is a vector. Now this isn't a math vector or a physics vector where it's like a pointy arrow that represents a force. This is sort of a little bit more like a disease vector. A disease vector, which we'll talk about later on when we cover epidemiology, is something that carries a disease from one person to another person. It's an intermediate. In this case, these vectors are going to be things which carry genes from one organism to another organism. So in transduction, the vector is a virus. Mostly, when we're talking recombinant DNA technology, the vector will be a plasmid. In fact, it's so common that the vector will be a plasmid that in the world of molecular biology, plasmid and vector are synonymous terms. When they say, so if somebody wants a vector and they say, hand me that plasmid or hand me that vector, they could be talking about the same thing. Now, there are natural plasmids and there are non-plasmid vectors, but those are the rarity. Those are the exception. Vectors need to be small enough that you can manipulate them in the lab, otherwise they're not very useful to us as humans. They need to be able to survive inside cells, since that's where they're going to end up. They have to contain what's called a recognizable genetic marker. And what that basically means is that these things are too small for you to see. You can't, like, look at the cell, see the vector, and go, yep, it got the vector. So you need to have something genetically that it's going to do. It needs to convey a new property to the cell that you can test for, so that you know that that cell actually has the vector. And last, it's responsible for the expression of a gene. So it has to be able to control the expression of a gene. And so a lot of the things that we've talked about before are going to kind of come together And you'll be able to see a technological expression of a lot of the stuff that we've talked about. So viral genomes can be vectors. They're often going to be called, I think, VACs, like viral artificial chromosomes, or phagemids sometimes. Transposons can be vectors, and plasmids. Usually it's plasmids. 9 out of 10 times it's plasmids. So if we're talking about a vector here, this is an example of a commercially available vector. And what it has is it has an origin of replication. Who remembers what an origin of replication is?
Where replication starts.
This is where replication starts, right? It's kind of in the name. If it doesn't have this, then the cell can't make a copy of this plasmid. The plasmid's dead in the water. But anything that has an origin of replication that's recognized by the cell's going to make copies of. Okay? It has, in this case, 2 markers. The most common marker used these days is an antibiotic resistance marker. This has been given resistance to 2 antibiotics, ampicillin and kanamycin. So you can just, boom, dump them on the plate, like dump some kanamycin on the plate. Everything that doesn't die got the plasmid. You can also use a positive selection, like sometimes there are ones that will glow. You guys are probably going to do a P-glow transformation in lab, which is a nifty plasmid that if the cell gets it and you express it, then you just look at it with a black light and the cells will glow green, which is kind of nifty. And there's 2 more important things here. First, this is called the multiple cloning site. It contains a bunch of different restriction enzyme palindromic sites all in a row. So you can take your pick. But these are what you're going to cut out. So what you're going to do is you're going to cut with, say, BAMH1 on this side, and HIMD3 on that side, and everything in between them is going to go away. You throw that away. Then you take your target DNA, and you cut your target DNA, like that's in, say, it's human insulin, right? You cut your target DNA with BAMH1 and HIMD3. It's got to be the same 2. Usually it has to be the same 2. Then you put them together, and the ends are sticky. So the BAMH1 sort of moving around here, or the target gene is moving around here, and then it kind of sticks there. And then eventually this is going to go and stick there. And you throw some ligase in, and it seals it up, and now you've got 1 piece of DNA with the new gene right in there in the multiple cloning site. And the last piece about this that's really important is this. So P-LAC means that this is the LAC promoter. You remember we talked about the LAC operon a while ago? And we said it has LAC-I, which is actually going to be around here somewhere in one of these unmarked parts. The LAC inhibitor is going to sit on there, but in the presence of lactose, it changes shape and then it falls off, and it's a negative inducible genetic system. What they did is they just cut that right out of the genome of E. coli, the lactose gene from E. coli, and plopped that promoter right in here. So you get this thing inside of a cell, and you add lactose, or actually what we usually use is xgal, which is a lactose mimic. Well, xgal is xgal is a different one. What we usually use is sorry, I have a brain fart, but it doesn't matter. It's not going to be in the test. I'll look it up later. What we use is a lactose mimic that doesn't get metabolized by the cell, but it looks like lactose. And as far as the cell's concerned, it says, oh, there's lactose. I should make this gene. And then whatever gene you put in here gets made. So that's restriction enzymes, and here's just a picture of that happening. You have your sticky ends. They're coming together. You ligate them. You transform it into a cell, usually using either a heat shock, which is what you guys will probably do, where you just dip your bacteria in hot water very briefly, and it's like the shock causes them to expand, and when they expand, it makes little holes in their membrane until it can catch up, and the plasma can sneak in through the holes. Or you can like give them a little electric shock, and the electric shock makes them vibrate so fast that, again, they get like little holes. All right, so PCR stands for the polymerase chain reaction. This is a way in vitro, not in a cell, of making a ton of copies of a piece of DNA, whatever piece of DNA you want. And this has been around since the 70s, but in the early to mid 80s, a big discovery was made, which allowed it to become much more popular, much easier to do. And that was the discovery of Taq polymerase. Taq is one of these, it's Thermophilus aqueous, and as you can guess from the name Thermophilus, it's one of those hot spring bacteria. And the problem with PCR is that there's a step in it where you have to get the water right up to almost boiling. And there are enzymes that it requires as well. So the problem was that you would get it up to almost boiling, and the enzymes would all die. And so you'd have to have a guy, usually a graduate student, come by and like add more enzymes in between every step. It didn't work very well. And Kerry Mullis, kind of a weird guy, did a lot of LSD at Berkeley, had like a vision one day. It was completely obvious in retrospect. And he says, hey, if we don't want the enzymes to like die when we boil them, why don't we take them from bacteria that live in boiling water? Their enzymes probably function at high temperatures. And everyone kind of collectively went, and it works great. And that's what we use to this day. I had a couple of videos I want to show you, because we're sort of low on time, and I think that the videos are going to be ultimately slightly better. Where is...
The polymerase chain reaction, or PCR, amplifies a specific DNA fragment from a complex mixture. First, the mixture is heated to separate the DNA strands. Two different specific oligonucleotide primers are added that are complementary to short sequence stretches on either side of the